Hello, everyone, and welcome to today's episode of our Discover Molecular Interactions webinar series. My name is Vivian. I am responsible for scientific communication at Dynamic Biosensors, and I will host this webinar episode today. So each month we look at a different molecular interaction and we discuss assays, how to characterize them, and we also explore what kind of insights you can gain from biophysical assays. And in this episode, we will talk about a specific mode of action that has come increasingly to the fore and that is influencing the conformation of a target protein. There aren't yet many technologies to measure these conformational changes in proteins, and so I am excited to be joined today by Pablo, a scientist from our R&D team, who will talk about how you can measure conformational changes using the switchSENSE technology. So hi, Pablo, welcome to the webinar.
Hi, Vivian. Thanks for having me.
So if you have any questions during the talk, please feel free to use the Q&A section, or if you can't see that, you can also use the normal chat function, and we will answer all your questions after the talk in the Q&A session. And during that, I can also unmute you and we can directly talk and discuss all your questions and comments. So now, without any further ado, I will hand over to you, Pablo, and I am very much looking forward to your talk.
Thanks, Vivian. So as she mentioned, there aren't very many technologies to detect conformational changes in real time. So I'm very excited today to talk about how we at Dynamic Biosensors use DNA origami nanolevers to actually detect protein conformational changes in real time. I'll start with a very brief introduction, and then we'll dive right into the measurement principle and how we use these DNA origami nanolevers to detect these conformational changes. And lastly, I'll finish with a few real examples that we've been working on in the past months. So I want to start by showing two seemingly unrelated structures. Here on the left, you have a petrol engine, and on the right, you have a human protein. And although these two structures seemingly have no similarities, what they actually have in common is that both of their functions arise from the coordinated movement of their component parts. And that's kind of the main message that I want to give today, is that proteins aren't these stagnant structures, but instead are these dynamic biomolecular machines whose function really arises through this interplay between different conformational states. At Dynamic Biosensors, we resolve the differences in these conformational states using the Helix adapter chip, which is integrated into our Helix Plus biosensor system. This chip is outfitted with 2 nanoelectrode spots to which we can functionalize these extensively customizable and electro-switchable DNA-based nanolevers. We can also take advantage of the integrated microfluidic system in these chips that's fully automated by the biosensor to, for example, flush in different analytes that bind to this ligand and induce a conformational change. This ligand is also customizable, and we can attach here any number of different proteins, even different peptides, and can even attach different RNA and DNA sequences. For the conformational change detection, we functionalize the surface of the chip on each of those nanoelectrode spots with 2 slightly different DNA-based origami nanolevers. On spot 1, we can direct our origami with our ligand of interest, in this case, for the purpose of the webinar, a protein that we suspect might undergo a conformational change. We can direct that origami every time to spot 1 using the complementary sequence of this anchor strand that we have fixed here onto the spot. Similarly, we can functionalize a DNA origami to serve as our reference origami nanolever that doesn't have a protein attached, for example. This serves as our reference origami so that we can measure both the conformational change occurring in this ligand and reference the measurement in real time. The reason we use DNA origami nanolevers is because they offer a few technical advantages to the standard adapter nanolevers that we use for different applications in switchSENSE. The main advantages are that the DNA origami nanolevers are much longer while still being quite slender and offer a very rigid structure. This means that we can discern even the smallest conformational changes that are occurring here in the protein on the distal end of the origami with a high degree of sensitivity. Moreover, because we have this densely packed bundle of DNA helices integrated into the DNA origami structure, we have a very high negative charge density. This means that we can measure the conformational change that we're interested in under physiological buffer conditions without diminishing the overall negative charge of that DNA origami nanolever. That negative charge is important because in practice, what this measurement actually looks like is that we functionalize the surface of the biochip with that DNA origami nanolever, as I explained, and apply an alternating voltage potential to effectively oscillate that DNA origami nanolever at high frequencies. When we apply a positive voltage potential to the sensor surface, we attract that negatively charged DNA origami nanolever to the sensor surface. As we do so, this tethered dye that we have here decreases in fluorescence because the dye is quenched by the sensor surface. So the closer we get, the more quenching we see. Then when we're in the fully down position, we can alternate the voltage potential to a negative voltage potential and essentially allow that DNA origami nanolever to diffuse back into the standing position. In doing so, we get a return back to baseline of the overall fluorescence of the system here. In this way, we can track the motion of the DNA origami nanolever as a function of fluorescence and repeat this oscillation for many, many times over the course of a measurement. We can then compile all of these motion curves together in what we call the average motion curve. And for a few technical reasons, we look primarily at the average upwards motion of that nanolever movement to discern relative differences in size of the protein that we're measuring and really make the basis for that conformational change detection. The idea and how we do this is by using Stokes' law for hydrodynamic friction, which states that the velocity of a particle is inversely related to its hydrodynamic radius. So what we see is that DNA nanolevers that are oscillating with smaller proteins on their distal end move much faster into the standing position than those DNA nanolevers with much larger proteins. This is how we can discern relative differences in the conformational state of the protein as well, before and after an analyte interacts with that protein, for example. What this looks like more technically in practice is that we look at the average upwards motion of those DNA origami nanolevers together and compare them with each other. So first, if we look at the reference origami nanolever coming back into the standing position, remember we get a return in the baseline fluorescence and we can track the motion of that origami like this. So the trace coming back into the standing position would follow more or less this line that you see here. We can then compare the movement of this reference origami nanolever to the motion of a theoretical nanolever with zero acting on it. So that theoretical nanolever, as we switch it back into the standing position, would move immediately up and stay up for the duration of the measurement because we have no friction acting on the system. What we can then do is compare the integrals of the two curves of the theoretical nanolever and of the reference real nanolever here. And what that looks like, if we compare the integral from time 2 to time 0 of that theoretical nanolever that goes immediately up and stays up, we would get an area of essentially this entire plot here. Then if we subtract away the integral from time 2 to time 0 of this reference nanolever, we would get this equation that you see here, and we represent that as what we call the instantaneous lag. In other words, how much the nanolever itself is lagging behind a perfect system. And this is represented by the shaded blue area here. We can then take a look at how the sample origami nanolever with our protein attached to it moves in relation to this reference nanolever. So that might look something like this. The first thing that you'll notice is that the movement of the sample nanolever with the protein attached moves a little bit slower into the standing position than the reference nanolever. In other words, the origami with the protein is lagging behind the reference nanolever. We can then quantify how much it's lagging behind the reference nanolever by, again, comparing the integrals of the two curves and generating what we call the sample-induced lag. So the sample-induced lag is the contribution that the protein itself is having to the overall movement of the DNA origami nanolever coming back into the standing position. Then, if this is a protein that we suspect undergoes a conformational change, we can inject the analyte that induces that conformational change and see how the upwards motion of the origami nanolever with the protein and the analyte changes in response to that analyte being injected. And what that might look like is a curve that looks something like this in green. So again, you'll see that in this case, the trace moves a little bit slower than both the origami with just the protein and the origami that we have for reference. Then, in order to quantify how much slower it's moving, we calculate what we call the dynamic lag change, which again boils down to essentially dividing this green area by the red area. And what the dynamic lag change tells us is essentially the isolated contribution that this analyte has to the overall friction of the system, specifically related to the conformational state of the protein here. Of course, in conformational change studies, a protein might not always move more slowly in coming back into the standing position as you see here. But what you actually might see is two different phenomena. So we can either have a scenario where we have the protein here and the analyte that we inject in causes either a compaction of the protein or an expansion of the protein. In the case of a compaction of the protein, there would be less friction acting on the entire system as that nanolever comes back into the standing position, and we would move a little bit faster than the nanolever with just the protein attached. So you'd see the average upwards motion curve of the origami with the protein shift in this direction here. Correspond to a negative dynamic lag change, indicating that there's less friction acting on the system, and we get a little bit faster movement of the DNA origami nanolever with the protein. In the case that I just showed, which is an expansion of the protein, we get an increase in the friction of the overall system and a shift in the curve in this direction, which corresponds to a positive dynamic lag change. So this is essentially how we quantify both the magnitude of the conformational change and especially in which direction that conformational change is occurring, either a compaction or an expansion. So now that we know how we calculate these dynamic lag changes in these proteins, I just want to show you a few examples of a few proteins that we've been working on in the past months, ranging from a variety of different research areas. So let's start first with the insulin receptor and then move to the other two examples. So in the unbound form, the insulin receptor adopts this open conformation as you see here. When we inject insulin into the system, however, you get this compaction of the insulin receptor that actually allows glucose to be transported into various tissues around the body. And this is exactly what we're seeing here when we run our experiments and calculate the dynamic lag change. So we get very consistently a negative 10% dynamic lag change, which again corresponds to a compaction of the protein, which is exactly what you see here from the crystallography data. Another very important part of these experiments is that we inject into the system an absence of the analyte that induces the conformational change. So we have a few runs where we inject only buffer. And as you see, in this case, we have no dynamic lag change, so corresponding to no conformational change in the protein, which is our integrated negative control into the experiment to make sure that the conformational changes that we're measuring, for example, in the presence of insulin, are true conformational changes and not an artifact of the measurement. In the next example, I'll show the maltose-maltodextrin binding protein where you see here labeled as MAL-E. Now this protein undergoes different conformational changes depending on the sugar substrate that's bound to it. So we can also generate functional data for the behavior of different proteins depending on the substrate that's binding to it and represent those in the dynamic lag change. So in this case, there are a few sugars that bind to the MAL-E complex and a few sugars that bind and get transported by this ATP binding cassette here. So what we did is first to inject just buffer into the system to make sure, again, that we have no conformational change when we just have buffer introduced into the system and we see no dynamic lag change, which is a good sign, means that our experiment is working as designed and we're not measuring any artifacts here. Then when we inject the sugar that should not bind this MAL-E protein and should not change its conformation, we get a very similar value here than we do in the case of just buffer, which matches very well to the functional data where this lactose sugar isn't binding to this MAL-E complex and causing this conformational change here. However, in the case of beta-cyclodextrin, this is a sugar that binds to the MAL-E complex, but then isn't in such a conformational state that it can actually induce this transport of the sugar into the bacterial cell. So in this case, we would see only a binding event that changes the conformation. And interestingly, that corresponds to a negative dynamic lag change value. But then if we look at these three sugars that you see here, maltose, maltotriose, and maltotetrose, they have a different functional behavior. So they not only bind to this MAL-E complex, but they also are able to be transported in by this ATP binding cassette here. The interesting part is that we get the exact opposite behavior. So we get a positive dynamic lag change for all three of these sugars, which lets us see that we can generate this functional data for the different substrates and how they interact with the MAL-E protein. The last example that I'll show is in this dihydrofolate reductase enzyme. Now, this enzyme is responsible for reducing dihydrofolate in the presence of an NADPH cofactor into tetrahydrofolate and is a very important player in nucleotide synthesis and has actually been shown to be overactive in a variety of different pathologies, such as rheumatoid arthritis and even some types of leukemia. And this drug, methotrexate, actually locks the protein in an intermediate conformation where it's slightly closed and actually prevents the activity of this enzyme. Under standard conditions, though, when dihydrofolate and NADPH are bound, this protein undergoes a conformational shift where the protein compacts quite a bit and binds that dihydrofolate very tightly in the active site that you see here. And this behavior can be represented also by the dynamic lag change values. So if we inject only methotrexate into the system, we get a compaction of the dihydrofolate reductase enzyme, which we see here again represented by a negative dynamic lag change value. However, when we inject NADPH, we see a much larger conformational change in the compaction of the protein, which is exactly what we expect from the data that's been measured previously in literature and corresponds very well to the story that we're building here. Very interestingly, when we combine both methotrexate and the NADPH cofactor, we get a conformational shift that corresponds mostly to what we see in the only methotrexate run. So this indicates that methotrexate is binding much more strongly to the enzyme than the NADPH, and the conformation that we're observing is actually this one, this intermediate conformation that the medication puts the protein into. So I hope with these few examples, I've been able to demonstrate the value of measuring conformational changes with these DNA origami nanolevers and demonstrate a little bit about how we do it here at Dynamic Biosensors. And of course, if you're interested in trying the DNA origami nanolevers and trying out our biosensor system, please reach out to us to inquire. The origamis are now in the web shop as well if you already have a biosensor and you would like to try these right away. And now, please, if you have any questions, I'm very happy to answer them. And also, if you have some questions in the future, please don't hesitate to reach out to me here at my email.
Thank you very much for this great talk, Pablo. And as you said, the floor is open for questions. There's already one in the chat that I will start with. So can you name a limit of detection with regard to the hydrodynamic radius? Is there a specific amount of change needed for this system to pick up on the conformational change?
Not necessarily. I'll say we haven't found the limit of detection. Of course, there probably is a limit of detection that exists, but one of the benefits of using the DNA origami nanolevers is because they're so long, even the smallest conformational shifts that happen in the protein here that we have on the distal end, we will be able to detect them if the conformational shift causes a change in the upwards motion of the origami. So if there's a change in friction, we'll be able to pick up on that. Of course, there surely is a limit, but like I said, we haven't come across exactly what that limit is quite yet.
Mm-hmm. So it would also be great to just test it. So if you have a molecule with this specific very small change, please also feel free to reach out to us, and we also arrange demos, for example, if you want to test first whether the system actually works for the proteins you are interested in. We can just give it a try first and see whether we can pick it up. Okay, I have a question. Not anymore? Okay. Okay, I will unmute you, and then you can directly ask your question. Hi. You should be able to unmute yourself now.
There you go. Can you hear me?
Yes.
Yes.
And so is there a way to use this technology to get some kind of a binding constants of a ligand to a protein?
Yeah, so we have, as I showed here, we have another technology where we can look at just the on and off rates of an analyte binding to whichever ligand you're interested in as well. This isn't done with the electrical actuation of the nanolever, so we're not applying this alternating voltage potential to the sensor surface, but instead we would apply a constant voltage potential that keeps the nanolever in the standing position, and then flush in the analyte that binds to the ligand here, and we're able to see the binding behavior because the binding event will change the local environment of the dye. And in that way, we'll be able to see how the analyte is binding to the ligand by this change in the fluorescence of the dye here. Then actually, we can also flush in buffer afterwards and also look at how the analyte unbinds from the ligand. So we can generate both the K on and the K off rates for that interaction.
So could you maybe send me information on that system?
I'm sure we could, yes.
Yes, for sure.
I have
for
my
sure.
you have my hand raised, so you know who that is. And then I guess from a technical standpoint, how do you get your protein attached to your DNA strand?
So we use a click chemistry reaction. So we pretty much attach this ligand strand here to a protein or peptide sequence using click chemistry. If we're going to use RNA and DNA sequences, we can of course synthesize those with the ligand strand extension here.
And so the click chemistry is with some kind of a reactive side chain of an amino acid on the exterior of the protein.
Right. So we can do a variety of different conjugations. So we can either do malamide coupling, amine, just standard amine coupling. So we really have a variety of different ways that we can do this, and it works quite well. We can also purify these conjugates using another device that actually I have here. The profiler. So basically you can make your conjugate and then inject it into the profiler and be able to select the fraction that comes out of the profiler that's a very pure protein DNA conjugate that you can then use in the biosensor.
Thank you.
Sure thing.
And for kinetics only, I think it's also an option to use tagged proteins. If you have a his tag or a GST tag on your protein, there could be an option for the kinetics assays too.
Exactly.
Okay. So the next question is whether we can use the method to identify an allosteric site. Have you done any experiments about this so far?
It's actually a very interesting question. So this protein that I showed here in the beginning is actually this ABL kinase protein, which has been shown to be overactive in certain types of leukemia as well. And this small molecule drug, imatinib, was initially this a great success story in drug discovery, but then there's been some resistance profile to this drug calling for allosteric binders. And the very unique part about our technology, I think, is that we don't have to look at an isolated binding pocket using our technology, because we use this difference in friction. So if we have the protein tethered to the origami nanolever, we're not limited to looking at only one binding pocket. So we can look at an analyte that, as you said, binds to an allosteric site and see the conformational change in that protein as a change in the overall friction of the system. So we're not looking at one part of the protein, but looking at the protein as a whole. So looking for allosteric binding sites is, I think, would fit very well also with the technology. Yes.
And can you also use this system for molecules that aren't specifically proteins? Or is this just for proteins?
Yes, I mean, in theory, we can customize the nanolever and the origami nanolever with whatever ligand we're interested in. We've done primarily proteins because many people are interested in protein conformational change, but I've also measured a couple of aptamers using the origamis, and we can also see in those aptamers conformational change occurring. The important part is that if there's a noticeable difference in how the origami switches with the protein attached or any other biomolecule, we'll be able to pick up on that. So if we have, like I said, an aptamer here instead of a protein, and the conformation changes when we flush in that analyte, which results in a change in the friction, we should be able to resolve that as well.
And what would you say are the next steps for your specific projects that you showed us before?
Sure. So in the maltose binding protein example, there are even more sugars that should bind and have this ability to be transported into the bacterial cell. So we'll expand the sugars that we're trying here to get this positive dynamic lag change. And there's also more sugars that only bind to the MOL-E complex. So we want to keep extending this story a bit and try all the known sugars that interact with this protein to see if we can get the full functional picture. And for the DHFR project, we just want to keep repeating these experiments and also expand the analytes that we're using to really nail down this story, because I think this one is also quite interesting. But we're constantly trying to increase the library of conformational changing proteins that we're using, and as the previous question just suggested, to really expand this to other biomolecules as well. So that's what you can expect next, and in the coming months.
Sounds great. I especially like how this one example really shows that this also has a functional consequence, the conformational change, and isn't just some structural, nice to know thing that we are measuring, but actually does something regarding the function in the cell. There was another question about publications. Whether we can send some publications. I will make sure that we send you some example publications via email, but you can also find all publications online on our website. There is a specific section on the website that is called e-learning center, and there you have a lot of filtering options, so you can find information like publications and former, like previous webinar episodes and app notes and so on, and you can filter also for molecule type and also for assay type. So if you go there, you can also find a lot of additional information. Okay, so I don't see any more questions at the moment. But as Pablo initially already said, please feel free to ask any questions also later on. by email or also through our website. There's also a contact form there. You can also contact us if you want to have a demo of the instrument at your labs, for example. And we also have our user meeting coming up. So if you would like to see more data on this, also on the kinetics assay, for example, there will be many speakers from, I think, 7 different countries. We have speakers until now. So a lot of scientists all over the world that will come to Munich this year in September and show us their data. So if you would like to participate in discussions with other groups, please also feel free to register for that. And with that, I think I will close this episode today. Thank you all very much for joining and for your interesting questions. And thank you, Pablo, for your talk and answering all the questions.
You're very welcome.
And I hope to see many of you soon in other activities or webinars. And I wish you a very nice rest of your day. Goodbye.